IB Bio, Option B, Biotechnology, Bioinformatics Part 10 is the second of two movies that looks at B.5 Bioinformatics. Part 10, B.5b, this movie will have its focus on applications of bioinformatics. This is the last movie for Option B. The essential idea is bioinformatics is the use of computers to analyze sequence data in biological research. Here is the outline of all the movies available for Option B. Use this outline to find the movie you need for review. The B.5 movies provide a very broad overview of the understandings in the IB syllabus. Most of the work to be done in this section of Option B needs to be done by engaging with the gene databases. Here is the first IB syllabus statement of this movie. Gene function can be studied using model organisms with similar sequences. Some organisms have been well studied. Drosophila melanogaster, the common fruit fly, this roundworm in the upper right corner, Mus musculus, the common lab mouse, and E. coli. These organisms will inevitably share genes or portions of genes with many other organisms. If the genomes of these organisms are completely known, they become the models upon which the study of less well-known organisms can be conducted. Some gene sequences have been conserved, not changed, among disparate groups of organisms. These sequences are important in metabolic pathways, and the comparison of genes between model organisms and less well-known organisms sheds light on the conserved sequences or mutations in the conserved sequences. In this diagram, you can see the conserved sequences among various mammal groups. If the conserved sequence is known in one organism, this organism becomes the living model for research involving other less well-known organisms. This diagram shows the percent amino acid identity among the various functional domains of the hepatocyte nuclear factor 4 protein. Numbers, here on the right, refer to amino acid residues. HNF4 is one of the most highly conserved nuclear receptors and is found in every animal organism examined thus far, from sponges to man. Viruses manipulate host cell biology through the expression of proteins that impacts the immune response of the host organism, the infected organism. Now, this diagram originates from scientists who hypothesized that the sequencing of viral communities would be useful to identify both known and unknown host immune proteins. To test this hypothesis, a mock human virome was generated and compared to the human proteome using the BLAST and search function in databases. This resulted in 36 proteins known to be involved in immunity. Now, the same process was then applied to reef-building corals, a non-model organism that currently lacks traditional molecular tools like transgenic animals or gene editing capabilities and in vitro cell cultures. So viromes isolated from corals and compared with the predicted coral proteome resulted in 159 coral proteins involved with immunity. Immunity is mostly studied in a few model organisms, leaving the majority of immune systems on the planet unexplored. Genome-scale metabolic models for gut microbiota are reconstructed based on different models and different databases. This image comes from research on seafloor microbes living in the muds below the seafloor. Many of the unique properties of the seafloor biota can best be investigated in the laboratory with model organisms. It is highly likely that subseafloor microorganisms harbor fundamental traits that differ significantly from those of their faster-growing, laboratory-adapted counterparts. Uncovering the biochemical and genetic bases of those differences will inform understanding of the fundamental physiological processes of sub-seafloor microbes. Specifically, the use of selected model organisms allows for the testing of these hypotheses. By studying model subsea floor organisms in the laboratory, one can gain important insights into physiological and metabolic features that are unique to the microbes in the subsea floor. So let's move on to the next IB syllabus statement. Use of knockout technology in mice to determine gene function. The mouse on the left has been genetically engineered to be unable to produce leptin. This results in obesity, and our understanding of the gene for leptin is enhanced with the use of knockout technology. A knockout mouse is a genetically modified mouse in which researchers have inactivated or knocked out an existing gene by replacing it or disrupting it with an artificial piece of DNA. You can see some of that technology shown in the image here. Knockout mice are important animal models for studying the role of genes which have been sequenced but whose functions have not been determined. By causing a specific gene to be inactive in the mouse and observing any differences from normal behavior or physiology, researchers can infer the probable function of the gene. The gene to be knocked out is isolated from a mouse gene library. Then a new DNA sequence is engineered, which is very similar to the original gene, except that it is changed sufficiently to make the gene inoperable, inactive, knocked out. Usually the new sequence is also given a marker gene, a gene that normal mice don't have that produces an observable change, like color or fluorescence. We've been through this before. The embryonic stem cells are isolated from a mouse blastocyst, a very young embryo, and grown in vitro in the lab. For this example, we will take stem cells from a white mouse. The new sequence is introduced into the stem cells by electroporation, and the stem cells are inserted into a growing embryo. Mice are widely used in knockout experiments, especially those investigating genetic questions that relate to human physiology. This is because mice are currently the laboratory animal species most closely related to humans. Knocking out the activity of a gene provides information about what that gene normally does. Humans share many genes with mice. Consequently, observing the characteristics of knockout mice gives researchers information that can be used to better understand how a similar gene may cause or contribute to disease in humans. The knockout technique provides researchers with a new tool for finding out what any given gene does. It allows them to genetically engineer a strain of mice with the gene missing or knocked out, and then watch to see what the mice can no longer do. After the first decoding of the mouse and human genomes in 2001, it yielded thousands of new genes of unknown function. Knockout mice became a prime source of information for making sense of these novel genes. Most human genes can also be studied in this way through their counterpart genes in the mouse. The mouse is a model organism, as about 95% of the genes in mice are conserved in humans. The first recorded knockout mouse was created by Mario Capecchi, Martin Evans, and Oliver Smithies in 1989, for which they were awarded the 2007 Nobel Prize in Physiology Medicine. Aspects of the technology for generating knockout mice and the mice themselves have been patented by private companies. Examples of research in which knockout mice have been useful include cancer, obesity, heart disease, diabetes, arthritis, anxiety, aging, and Parkinson's disease. So let's move to our next IB syllabus statement. Sequence alignment software allows comparison of sequences from different organisms. You should remember from Part 9 that BLAST-N allows nucleotide sequence alignment, while BLAST-P allows protein alignment. Sequence alignment describes the way of aligning DNA, RNA, or amino acid sequences to highlight or identify similarities between the sequences. Typically, gaps have to be inserted into sequences so that identical or similar nucleotides or amino acids are aligned in columns, as you can see in the example shown in this slide. The technique involves isolating DNA from each of the species studied, generating DNA sequences for a gene region, an entire gene, or several genes, assemble the DNA sequences for each species into separate quote-unquote contigs, align the contig sequences to each other using a multiple sequence alignment program, software program such as Clustal W. And then you verify any observed differences by going back to the original DNA sequences. Base-by-base alignment of one sequence to another allows for similarities, mismatches, and gaps. Here's the example. Two sequences. Computer software performs the alignment, allowing for similarities, gaps, and mismatches. As you can see here, these two sequences are 80% similar. More detailed sequence similarity analysis can be performed by creating multiple sequence alignments. By this method, 3 or more biological sequences, amino acids or nucleic acids, similar lengths are aligned. They're aligned to minimize gaps and maximize the occurrence of identical or similar residues at the aligned positions. From the output alignments done by computer software, homology, in other words, evolutionary relationships between the sequences, can be inferred. Moreover, the presence of conserved regions indicates conserved structural and functional elements within the sequence. The most often used program for multiple sequence alignment is Clustal W, using this URL. On this slide, 3 polypeptide chains from the Globin family are aligned by the Clustal W software program. The asterisks at the bottom line of the alignment indicate identical, fully conserved residues in a given sequence position, while single and double dots refer to highly and moderately conserved residues, respectively. Within the aligned sequences, the dashes indicate the gaps that were inserted in order to optimize the alignment. And that leads us directly to this IB syllabus statement. Multiple sequence alignment is used in the study of phylogenetics, and use software to align two proteins. Clustal W is the software of choice. But before going too far, too fast, let's define phylogenetics to ground our understanding. Phylogenetics is the study of the evolutionary history, evolutionary relationships among individuals or groups of organisms, species, populations. Take some notes. Base or amino acid sequences for common genes can be aligned for different species. Alignment allows for comparison. Computer software allows the comparison to be analyzed for phylogenetic relationships, evolutionary relationships among the groups of organisms seen here. In this slide, we see amino acid alignment for a specific protein in different species. The amino acids are lettered in FASTA format. Multiple sequence alignment is used in the study of phylogenetics, and using software to construct simple cladograms and phylograms of related organisms using these DNA sequences. Here we can see, once again, multiple sequence alignment for different species. Now, look carefully. Based on the alignment, can you see any evolutionary relationships among these species? Here's the cladogram that results from the multiple sequence alignment. You can see that the common ancestor of the human and gorilla lived more recently than the common ancestor of the human and the green monkey. Thus, the human and the gorilla are more closely related than the human is to the green monkey or the gorilla to the green monkey. Here's the multiple sequence alignment for subspecies of 3 species of Boletus fungus. And here's the cladogram displaying evolutionary relationships. Once again, here's the multiple sequence alignment, and here's the cladogram displaying phylogenetic relationships. And that brings us to the last IB syllabus statement of the movie. Explore chromosome 21 in databases using software ensemble. To a large extent, this is an exploration that you can do independently. You should remember that chromosome 21 is a very small chromosome, and you should remember that a relatively common non-disjunction event in the production of an egg in older females results in trisomy 21 following fertilization. For the exploration of chromosome 21, go to this URL, select human in an effort to explore chromosome 21. You'll come to this page, select View Karyotype, and you'll find this page that displays the 22 chromosomes and the X and the Y. Notice that chromosome 21 is the smallest chromosome of the human karyotype. Click on 21 and notice an option, choose Chromosome Summary. And that brings you to this page. There's a lot of detail here, but just for a second, notice that you could explore any chromosome of the human genome by clicking here. And then you can explore by clicking on any region. This looks interesting because there are numerous protein coding genes in this region of the 21st chromosome. And that brings us to this page, and you can see that there's a lot of detail. We have drilled down to this section of the chromosome. You can scroll back and forth along the chromosome, and notice that the genes are color-coded. Here we have genes that code for RNA molecules, such as ribosomal RNA or transfer RNA. Let's back out a layer, back to the chromosome summary slide, and note that just below the chromosome summary are chromosome statistics. Total length and base pairs of chromosome 21 are given, and the total number of known genes is given. If you back out one more level to the whole genome page for humans, you can see the total number of base pairs in the human genome at roughly 3.5 billion. You can see the number of identified genes at just over 20,000. And at the top of the whole genome page, you have the option of investigating the genome of many, many different species. And that brings us to the end of IB Bio Option B, Biotechnology, Bioinformatics, Part 10.